Okay, hello everybody. So this is the Mitosis and Meiosis lecture from the Level 1 Human Genetics module, part of the Biomedical Sciences degree at the University of Bradford. So what we're going to be talking about today are, well, it says here mitosis versus meiosis, as though we have some kind of Harry Hill fight-off plan for the two different things, but I guess it's more of a comparison between mitosis and meiosis that we're going to be concentrating on. The two processes share a lot of similarities, but there are key differences which we're going to cover today in some detail. So we're going to talk at length about mitotic nuclear division, including a little bit of information about the actual apparatus which exists in the cell in order for that process to occur, the mitotic spindle. Consider the question how chromosomes move around on this apparatus during mitosis. And then we're going to think a little bit about how another process very similar to mitosis called meiosis occurs, why it's important and how it differs from mitosis, including a little bit of information about haploid and diploid life cycles, and look at what a meiotic nuclear division looks like, and then consider how meiotic nuclear division can generate variability, which is obviously a key aspect of the process. That's why the process exists, and finish with a summary of the lecture. So it's going to be hopefully quite a short lecture today, around 40 minutes or so. Okay, so what we've got here is just a few comparisons really of the two processes and some differences, just in table form, just for your convenience later on when you're revising, and just to get us thinking about the two different processes at the top of the lecture. So mitosis then you can think of as the division which actually occurs in the vast majority, in fact all bar one cell type in your body. So every cell that's divided in your body is undergoing mitotic nuclear division, with the exception of one cell type, which is the sex cells or gametes. So these cell types are generated via meiotic nuclear divisions. So there's, from the top we can see that meiosis is a very specialised process. It's not going on all over the place, it only goes on in one particular cell type in the body. Mitosis is non-reductive, so the chromosome number remains the same before and after the division process. Meiosis is a reductive division, so that's to say that once meiosis is finished, you end up with half the number of chromosomes that you started with. Mitosis occurs over one nuclear division, one division of the nuclear material. Meiosis involves two consecutive divisions of the nuclear material. So if you think about genome number and mitosis, what we have is we have one N to start with, one genome that is replicated, and this is a typo, it should actually say 1N at the end. Whereas for meiosis, we start off with a 2N with a replicated genome, and then the cells which we eventually produce at the end of meiosis actually only have 1N, so we've lost half the chromosome number. So mitosis then, let's consider this first in a bit more detail. Well, it's been studied by cellular biologists for over a century, mitosis, but it's really only the last couple of decades, with the advent of a much stronger understanding of molecular biology, that the mechanisms that control it have really began to be unpicked by scientists. So it's been studied for so long because it's such a visible process. Mitosis. It's very easy to see via microscopy. There are huge changes, as we'll see in a moment, going on in the cell, which are very easy to see even with quite basic light microscopy. So it just so happened that the process itself was very easy to observe, but it's not been very easy to unpick and understand at the molecular level. So the process occurs during M phase, which we've talked about in previous lectures on cell cycle, and effectively M phase, or the process of mitosis, is composed of 5 separate stages, which you may have heard of already at A level, I guess. So these are prophase, prometaphase, metaphase, anaphase and telophase. So a bit of a mouthful, some of those names, but they're the 5 main stages that make up mitosis. So we'll go through what each of those encompass over the next few slides. So before we do that, I just want to show you what the cell looks like prior to the onset of mitosis. So this is a G2 a cell that's in G2 phase of the cell cycle. So if you remember from the cell cycle, G1 is a growth phase, S phase is when we replicate our DNA. Following DNA replication, there's a short secondary growth phase where the cell gets ready for mitosis, and this is what we're looking at here, this G2 phase cell. So if we look at a G2 phase cell, we can see we have an intact nucleus still bound in the nuclear membrane. This is a chromatin inside the nucleus that's been duplicated. So this is a cell which has a genome number of 2N. It's undergone DNA synthesis. And then we've got these structures here, so-called centrosomes or centrioles, and these are actually what are called mitotic spindle organizing complexes or organizing centers. And it's from these centrosomes that the mitotic spindle apparatus is going to emerge during mitosis. So let's have a think about prophase then. So several things occur during prophase. The first thing to occur is that the chromosomes start to condense. So if we look at the nucleus now, we've no longer got this interface chromatin which just looks like a nondescript mass of DNA. We start to see these more familiar chromosome shaped pieces of DNA emerging in the nucleus. These are replicated sister chromatids. So this is a chromosome, remember, that's undergone DNA synthesis. It's still attached to its newly synthesized sister chromosome here. See these microtubule organizing centers now starting to emit these mitotic spindles. So this is a mitotic spindle apparatus which is going to be used by the chromosomes during mitosis. And these mitotic spindle organizing centers move away from one another to opposite sides, opposite poles of the cell, ready to form a complete mitotic spindle apparatus in the next phase of the cell, next phase of mitosis. So the next phase is prometaphase. So in prometaphase, the first thing to happen is that the nuclear membrane breaks down. So we've no longer now got a membrane-bound nucleus in the cell. The nuclear membrane is dissolved, and the mitotic spindle then invades inside this intranuclear space, forming a more complete mitotic spindle apparatus. As this happens, the spindles themselves actually seek out and attach to a region called the kinetochore of the chromosome. I'm going to talk more about that later on. So these spindles hook onto the chromosomes. There are actually three types of microtubule that are now present in the cell. There's a kinetochore attached microtubules, polar microtubules which are invaded into the nuclear space, and these astral microtubules which are found facing away from this intranuclear space. So the next stage of the cell cycle is a very important stage called metaphase. So in metaphase, the chromosomes are fully condensed. So it's in metaphase cells really that it was metaphase cells really that the first early the first early scientists who were using basic microscopes were able to observe the process of mitosis by looking at cells which are in metaphase. That's simply because the chromosomes in metaphase are the easiest to see out of all points of the cell cycle. They become very condensed and look like the cartoon interpretation of what a chromosome looks like. So in metaphase, in addition to the chromosomes becoming very condensed, they also begin to align on something called the metaphase plate. So the metaphase plate, if you like, represents the mid-body, the centre of the cell, as directed by the presence of this mitotic spindle network here. So because the chromosomes are most condensed in metaphase, it's during metaphase that you actually carry out something called karyotyping analysis, which again I presume you've heard of during A-levels, but if not, karyotyping is what cytogeneticists look at in order to identify the presence of certain chromosome translocations or aneuploidy, so the presence of a third chromosome 21 for suspected Down syndrome cases, etc. What happens is that cells are isolated that are in metaphase, and the chromosomes from those metaphase cells are spread out onto a microscope slide. The chromosomal DNA is stained by using something called chiasma staining, which causes the chromosomes to band, do take on a particular banding pattern, and cytogeneticists can look at this banding pattern and then interpret that information to determine whether or not the chromosomes are normal, inverted commas, or whether certain translocation events might have occurred, or chromosome breaks, etc. Anyway, back to the metaphase cell. During metaphase, once these chromosomes have aligned on a metaphase plate, they're actually under a fair degree of tension. So the chromosomes are kind of held in a state of tension at the metaphase plate via the actions and opposing actions of the microtubules, which are attached to the chromosomes. So the next stage is perhaps the most dramatic stage of mitosis, that's anaphase. So during anaphase, several things happen again. It's the shortest phase of mitosis, that's just because it happens so rapidly. And what happens once anaphase is licensed, or once the cell receives a signal to begin anaphase, replicated sister chromatids suddenly fly apart from one another and move towards opposite spindle poles, opposite microtubule organizing centers here. The first stage of anaphase, anaphase A, chromosomes are pulled towards the mitotic spindle poles by shortening of the kinetochore microtubules, so these microtubules which are attached to the kinetochore region of the chromosome. And then in anaphase B, the chromosomes are separated further as the spindle poles themselves start to move away from one another. So it's kind of a two-step process, but it happens very rapidly, as you'll see in a video hopefully later on. And the final stage of the cell cycle is telophase. So in telophase or telophase, the replicated chromosomes arrive at the spindle pole. The chromosomes unwind, start to reform this more dispersed chromatin structure that we see in interphase nuclei. The nucleoli of the nuclei begin to reform, as does the nuclear membrane. So we start to see the presence of a membrane-bound nucleus again. And finally, cytokinesis occurs. So cytokinesis is the final stage of the cell cycle, and this is where the two cells are actually fully separated cytoplasmically. This occurs via the action of a actin-myosin band that you can think of pretty much like a belt that tightens around the midriff of the cell here until such a point that there's a very small waist, if you like, left here that's then pinched, and you end up with two separate cells. So that's the process of mitosis, really, in a bit of a whistle-top stop of the process. So what we want to think about now in a bit more detail is the actual mechanics, if you like, and the apparatus which a cell possesses and uses to carry out mitosis. So the first thing I want to talk about very briefly is this mitotic spindle structure. So the mitotic spindle looks like this if you stain for tubulin, which is a major component of the spindle apparatus. This is a cell that's actually ready to, or probably just undergone anaphase. You can see it's got a fully formed mitotic spindle network here, reaching into the intranuclear space. There's fewer microtubules here along what is the metaphase plate, and you can see the spindle organizing centers here and here with the astral microtubules spinning out from it. This is shown here in this cartoon. It's the same thing. What you can't see here because they're not stained is the chromosomes, but in the cartoon you can see how the chromosomes here are lined up along the metaphase plate. So all of this microtubule network is composed primarily of a protein called tubulin. And tubulin is clever because what the cell can do with it is much like adding it's just like Lego bricks or blocks together. The cell can add and remove tubulin subunits, and it's this addition of tubulin subunits that allows these spindles to initially form and these microfilaments to invade and move into the intranuclear space. So quite simply, when the addition of tubulin subunits outstrips the removal of tubulin subunits, then you get positive growth of the microfilaments. In other words, if the blocks that are getting added on, if that happens at a rate that outstrips the removal of those blocks, then the microtubule will grow. Conversely, if the blocks are removed more rapidly than they can be added, then the microtubule will shrink. And that's how the cell deals with either extending or reducing the length of these microtubules in the cell. So how do these mitotic spindles actually get hold of the chromosomes then? So I mentioned earlier that they actually kind of latch onto them. Well, the chromosomes actually contain a specific structure towards the middle of the chromosome, the centromere of the chromosome, called a kinetochore. And the kinetochore is the structure to which these tubular microfilaments bind. So kinetochores form what's called a centromere of the chromosome, and it's been shown that centromeres contain a particular DNA repeat sequence that we believe is important to the actual formation of a very condensed type of chromatin at that particular structure. And likewise, this formation of this particular dense chromatin structure at the centromere and at the kinetochore then facilitates the latching on of tubulin via the so-called Fisherman hypothesis. Fisherman hypothesis is very straightforward. This is the hypothesis that the microtubules kind of just pitch inwards into the intranuclear space almost at random until they latch onto a kinetochore region of the chromosomes, and when they actually manage to latch onto the kinetochore, then if you like, they hook the fish, they hook the chromosome, and then we're ready to start aligning these chromosome structures on the metaphase plate prior to anaphase. So we've hooked our chromosome. How do we reel it in? Well, effectively the mitotic spindles actually pull the chromosomes apart. But it's an interesting phenomenon that whilst mitotic spindles are pulling chromosomes towards the daughter cell, towards the spindle pole, mitotic organizing center or the spindle poles themselves actually repel the chromosomes. So we've got an interesting situation where we've got two different opposing forces at work, and this diametric force, these diametric forces basically explain how the chromosomes come to be aligned on this metaphase plate structure. So if you imagine a situation where the spindle poles are repelling the chromosomes and at the same time the microtubules are trying to pull them, then what you eventually reach is an equilibrium whereby the DNA settles in the centre of the cell. So it's equally distant from either spindle pole, but at the same time it's desperately being tried to be pulled towards those spindle poles, so it causes the chromosomes to just settle at this metaphase plate region. We know that this is how it works via a series of experiments. One particular elegant one is one done with laser microdissection of cells undergoing mitosis. And what the scientists basically did here is they took a mitotic cell that was in the early stages of anaphase, so this is where chromosomes were starting to move towards the spindle pole bodies, and they severed the top half of the chromosome above the kinetochore and showed that those chromosome regions which were severed and no longer attached to the kinetochore were actually flung away from the spindle pole body. Taking this is evidence that the spindle poles are repelling the chromosomes all the time, and it's only the action of a greatly shrinking or decreasing microtubule apparatus here that's attached to the kinetochore that pulls the chromosome towards it at a rate that's more powerful, if you like, than the repulsion going on from the spindle pole body. So I've painted maybe a little bit of an oversimplistic picture thus far of how these things are moving along. It's not. I've maybe give you the implication that it's a passive process, simply the microtubules just shrink and the chromosomes move with them, but the process is actually a lot more active than that and involves the action of a family of proteins called motor proteins that are actually responsible for dragging the chromosomes apart during the anaphase stage of the cell cycle of mitosis. So the major protein involved in moving chromosomal DNA along microtubules is a motor protein called kinesin, and it's the same protein that's involved in moving chromosomes around in mitosis and meiosis. So kinesin works or can function as a motor protein due to its ability to be able to walk along microtubules whilst attached to cargo. Cargo in this case is chromosomes. And this is a cartoon, if you like, or a diagram of what the kinesin protein looks like and how it actually works. So this is a tubulin, tubulin microfilament, so part of the mitotic spindle network. What you have here are dimers of tubulin, alpha and beta subunits that stack up together to form this long microfilament. And then what we have here is kinesin, and what kinesin does is it interacts with this microtubule filament, but one subunit at a time. So if one part of the kinesin protein is bound to microtubules, the other part isn't. And in an ATP dependent manner, so an ATP driven reaction, you get phosphorylation of this non-bound subunit, which causes the entire protein then to rotate and this subunit to bind to the tubulin microfilament as well as the other subunit, at which point the other subunit lets go. And what you effectively create by this ATP driven mechanism is a kind of walking protein, a protein that can kind of rotate and walk along the length of the microtubule filament. So in the lecture I may well mimic this walk to almost an uncanny level of accuracy. In the lecture theatre I can't do that via a podcast, but you want to imagine this is rotating round in such a way that it's kind of spinning and walking along the microtubule filament. So what we actually have going on in the cells then is this situation where we've got these great highways of microtubule filaments, to which we have many molecules of the kinesin protein motor, which is dragging with it the chromosomal material during mitosis. So it's like a delivery system, it's like FedEx for the cell, this kinesin. So once you've separated the chromosomes and you've ended, you're ready to end mitosis, end the cell cycle. And the final stage of the cell cycle is cytokinesis. So this rather unpleasant picture here is actually an animal cell that's about to undergo cytokinesis. An early event in cytokinesis is the formation of this so-called cleavage furrow. And this is formed by the action of an actin-myosin ring. So this is a ring or a belt, if you like, of actin and myosin, which are two other types of motor proteins and structural proteins. Very similar in a lot of ways to tubulin and kinesin. Actin is very similar to tubulin, that it's a structural protein. Myosin is very similar to kinesin in that it's a motor protein. And what happens with actin and myosin is that you form this belt structure around the cell, and then this belt is effectively tightened until such a point is reached that the two cells are completely separated, and you end up with two brand new daughter cells being formed. So myosin, you may have met the leap with muscles, muscle tissue, and it's just the same deal, this actin-myosin ring, as how striated muscle works, how it can slide over one another to contract when the muscle tightens. So the same way this belt tightens, this belt of actin and myosin, it's very similar mechanically to the tightening of striated muscle, just in standard tissues. So what we've got here is this is some immunofluorescence staining, rather nice immunofluorescence staining of a cell that's looking at actin and myosin and then tubulin. So this is the tubulin here in orange. You can see the remnants of the mitotic spindle that's been leading into the intranuclear space, the spindle organising centres here. The blue is actually actin, so you can see how actin looks very similar to the tubulin in some ways. It's a structural protein. It's concentrated here at this point, which is all that's left really of the middle of the cell after the actin-myosin ring has fully constricted, and this cell's obviously about to complete the cell cycle and divide into two new daughter cells that will then enter G1 of the cell cycle. So that's mitosis and some of the mechanics that are controlling it. So what about meiosis then? So a couple of important things that make meiosis different from mitosis. You know, often if lecturers put things in a different colour on the slides, it's a good idea to make sure you understand these terms and remember them, because they're obviously important, one, to understand the topic, but two, may be involved in important during assessments as well. So meiosis then involves one round of DNA replication and 2 rounds of nuclear division. So in other words, you don't get more DNA synthesis going on, you just get more rounds of nuclear division, which is why it's a reductive process. So you replicate your chromosomes once, but then you divide them twice. So rather than going from one end to 2 end, and then back to 1 end, you start off with 2N in mitosis and you end up with 1N. You lose half the chromosome number. So in addition to generating the gametes required for sexual reproduction, meiosis is also a source of genetic recombination. So we already mentioned earlier on that the gametes, the sex cells, are produced via meiosis, and it's also meiosis which allows effectively the reorganisation or the jumbling up and recombination of genetic information during sexual reproduction, which is obviously a key underpinning of the generation of genetic variation, the evolution of the species. So we're talking about genetic variation in the next lecture. So meiosis then, with regards to the actual stages of meiosis, like mitosis, it's divided into numerous stages. Most of these are exactly the same as mitosis. So once you've learnt mitosis, the good news is you already kind of know meiosis. So most of the stages are identical to mitosis, with the exception of prophase 1. So this is the additional bit that you need to learn and understand for meiotic divisions. So prophase 1 is a very long and complex part of meiotic division that can take a long time to complete. And it's that complex and that involved that it's been subdivided again into more stages. So you've got even more silly names to remember, I'm afraid. Greek and Latin derivatives. So leptotene, zygotene, pachytene, diplotene and diakinesis form prophase 1. So let's look in more detail at these stages like them, what's going on. Before we do that, what I've got here is an image of a chromosome that's just undergone DNA synthesis and is now undergoing genetic recombination. These are chiasmata, touching points between sister replicated chromosomes, and we'll talk about them in more detail over the next few slides. So during leptotene then, the chromosomes condense, just like during prophase and metaphase in mitosis. So leptotene, you get more chromosome condensation. Zygotene, further chromosome condensation as well. But importantly, it's during zygotene that you first start to actually pair up homologous chromosomes to form what are called bivalents. So obviously, if you take any cell from your body apart from a sex cell, which is a haploid cell, the cells in your body are diploid. In other words, each cell contains a chromosome from your mum and a chromosome from your dad. 46 chromosomes, 22 chromosome pairs, an X and a Y. During meiosis, during meiotic division, once those chromosomes have replicated, what happens is these homologous chromosomes pair to form these bivalent structures. So the chromosome 1 from your mum will find the chromosome 1 from your dad and will pair up with it. Then during pachytene, we get the event of crossing over occurring between bivalents. And it's this crossing over during pachytene which generates genetic recombination. We'll look at that in more detail later on. But this is a key aspect of meiosis. The ability to share genetic information or swap genetic information with a chromosome sister, with a homologous chromosome. Diplotene, you start getting the separation of these homologous chromosomes, but they actually stay touching to one another for a fair while yet at points which are called chiasmata. And then diakinesis, the homologous chromosomes now are really moving quite far apart. They're about to be completely separated, and it's only the chiasmata, these touching points now, that are actually holding the homologous chromosomes together. So you can see these homologous chromosomes being slowly pulled apart, but at several points you get these points where the chromosomes touch, and this is the so-called chiasmata and points of chromosome recombination. So the key difference between meiosis and mitosis then is the fact that you get homologous chromosome pairs aligning with one another at the metaphase plate in metaphase 1. So this is a nice diagram to some of the differences between the two processes, because it also shows the similarities. So for example, everything starts off the same. You have a paternal and a maternal chromosome that undergoes replication during DNA synthesis. So each of these chromosomes then is attached to a chromosome sister. The difference then is that in meiosis, these two chromosome pairs, this chromosome pair will align next to one another and cross over and maybe even share or swap genetic information prior to dividing away from one another during metaphase 1. Then you will get a second round of cell division occurring, and this time now you will get separation of the chromosomes in a way that's similar to the separation that occurs during mitosis. But because you've already undergone one nuclear division, when you end up with the final cells here at the end of meiosis, each of these cells, each of the gametes, only contains one chromosome. It doesn't contain a chromosome pair as we see here in the mitotic nuclear division. And also importantly, some of these chromosomes may have undergone recombination events during meiosis to generate in effect a new chromosome that's genetically different from either parent. So it's the pairing of homologous chromosomes during prophase 1 and the exchange of genetic information that's really the key aspect of meiosis. That's the thing you really need to understand with regards to why it's important. So how does this crossing over during packaging generate genetic variability then? Well, let's consider this diagram to start with. This is just another example of what's going on during meiosis. And here we don't actually get crossing over the chromosomes, but we do get different combinations of gametes being produced depending on how the chromosomes pair during the meiosis 1 and meiosis 2 stages of meiotic division. And you can get a mixture of gametes depending on whether or not the maternal and paternal chromosomes segregate together or with one another, as you can see in these different diagrams here. It's this diagram that's particularly important though, because what we see here is the effect of actually genetic recombination occurring. So this is crossing over of chromosomal material. So if you have two different traits, two different loci on the same chromosome, A and B, and we have a chromosome pair here, and on chromosome, the red chromosome, we have, if you like, dominant big A, big B alleles for these two loci, and on the blue chromosome, we have recessive little a, little b alleles for these two loci. Were no crossing over to occur, the only gametes we would expect to be able to produce from these starting materials would be big A, big B, little b. However, if you are able to get crossing over occurring between these two chromosomes during packaging stage, what can happen is you can get a swapping of the material from the blue chromosome with the material from the red chromosome, like we see here. So the bottom arm of this chromosome has crossed over during packaging and has been resolved in such a way that we have swapped the information between these two arms of the chromosome. And then what happens during the second meiotic division is that you actually go on to generate these gametes, big A, big B, little b, but also these recombinant genotypes of big A, little a, big B. And it's this mixing up of genetic information, this genetic recombination, that's really key to meiosis and key to the evolution of sexually reproducing species, because we have the ability to generate almost a limitless variability within our genome, as we'll discuss in the next lecture in the module. So how does this sharing, this recombination occur then at the molecular level? What's going on? Well, this is quite a complex process involving something called a holiday junction, and I've tried to summarise it here, but you don't need to worry about any more detail on what this slide shows, although you will be able to find more information on this in the core textbooks. What occurs between the two chromosomes here is, as the chromosomes are paired up, a double strand break event occurs in one of the chromosomes, and what that then allows is for one of these free ends of this broken chromosomal strand to invade the homologous chromosome region here, and that displaces this other strand in the unbroken chromosome in such a way that it acts as a template then for the repair of the broken strand. But because this chromosome here is acting as a template, the repair that occurs using the sequence that's present in the undamaged parental chromosome. So when this repair mechanism completes and that damage is resolved, what you find is that you end up sharing information between the two chromosomal pairs. Now, depending on how this repair is resolved, depends on what type of crossover event you get. So you've got to remember that chromosomes are 3D structures. It's actually more useful to think of them like this when they're paired up, as opposed to these linear diagrams. And if you think of them like this and think about where in these double-stranded chromosome structures we need to cut the DNA in order to resolve the repair, depending on where that cut is made, we will get different types of genetic recombination. So if the cut occurs in this plane, we'll generate this type of crossover event, whereas if the cut occurs in the horizontal plane, we'll generate a very different type of crossover event. But in both instances, we are sharing and mixing the genetic information between one chromosome and the other. So you may need to go away and read a little bit more about this in the additional reading before this sinks in. But it's a very important process, and it's quite important you understand and can get your head around the spatial aspects of this. It's quite difficult to get across in a podcasted lecture. So to summarise then, I told you it was a relatively short one this week. Meiosis is the process of nuclear division that generates the sex cells, the gametes. Mitosis produces everything else. Mitosis just has one round of DNA replication and one round of nuclear division. So you double your number and then you half it again, so you get back to where you started. In meiosis, you have one round of DNA replication, but 2 rounds of nuclear division. So you end up with half the number that you started with. You don't get genetic recombination during mitosis because the chromosome pairs never pair up like they do in meiosis. In meiosis, during prophase 1, you do get homologous recombination. Homologous chromosomes forming pairs, forming these bivalent structures. And once they're in a bivalent structure, chromosomes can share and exchange genetic information by crossing over at specific points, which are called chiasmata. When these chiasmata crossover events occur, it causes a particular structure to form between the homologous chromosomes, and this structure is called a holiday junction. So, thanks for your attention. I hope the last couple of slides there weren't too unclear. You may, as I've said, need to go and look at the core reading material before that kind of sinks in, particularly about the holiday junctions and resolving of different planes during crossing over. But the main things I want you to take away from this lecture are a really clear understanding of the different stages of mitosis, and also the key differences between mitosis and meiosis. Yeah. So if you've got that clear in your head, then that's you're a long way on the way there to meeting the learning outcomes of the lecture. Okay, so listen up for more podcasted lectures in this module, and I hope this proves useful during revision when you revisit the material. Thank you.